3:30, which is our starting time. My name's Carol Knight, and I'm the head of campus here at Duchy Rosewarne. But I've got the wonderful task of introducing this afternoon's talk, which is entitled Cornwall's Raucous Summer, Ecological Restoration for Conservation Optimism. And this is being delivered today by the wonderful Mark Nason. Mark is a certified ecological restoration practitioner, the academic lead and programme manager of the Master's Land and Ecological Restoration course at Eden. He's also head of Eden Project Learning. That's his new title as well, which is congratulations. Eden Project Learning offers a specialist portfolio of higher education courses ranging from plant science to sustainable festival management. To learn more, please visit the Eden Project Learning website, and so you might be keen to do that. So I'm just, before we start, just to say, if while we're in the session, if you can keep your microphones off, because we do sometimes get a bit of you'll have sometimes come across echoing, so if you can keep your microphones off, and if anybody would like to ask any questions either during the session or probably what would you prefer, Mark? Would you like it to be at the end or during the session? Or
I'm easy. Maybe put questions during in the chat box, because it might be that a couple of my colleagues here could answer them as well.
Okay, fantastic. So yes, if anybody's got any questions, if you'd like to put them in the chat box, then that's fine. If you want to put your hand up at the end and ask anything, there'll be an opportunity to ask questions at the end. But in the meantime, the session is being recorded, so you are all being recorded, and this will be available through Planet eStream for you to access afterwards as well. So I will hand over to Mark. And we are very interested to hear all about Cornwall's raucous summer.
Okay, thank you very much, Carol. So I'm going to share my screen. So hopefully you can all see some slides now. And I'll keep this relatively informal, and I'm going to try not to talk for too long, because I also have two of our students here, and they're able to tell you about the projects that they're working on over the summer. So the title, the Raucous Summer, is plagiarized directly from George Monbiot, and he talked about the need for a raucous summer of restoration and rewilding, and how that is the antidote to the silent spring that we've been experiencing with the collapse of our biodiversity, that was first highlighted by Rachel Carson in the 1960s. And so we are planning a raucous summer, and actually it's a summer that's going to last 10 years is the plan, and I'll tell you more about the 10-year plan during the presentation. But also we are running an MSc in Ecological Restoration at Eden, and we're in the second year. The first year of the programme, we launched it during the COVID year of lockdowns, and we were unable to complete any of the practical research projects, the field and lab-based projects during the summer because of lockdown. So this year we are really thankful and really excited to be able to get out and about with 15 field and lab-based practical projects running until the end of August. So as well as needing a raucous summer as an antidote to the silent spring, this is also our raucous summer of field work, and we're going to make the most of it between now and the end of August. So I am head of campus at Eden Project Learning, as Carol said, and my background before moving into education was research into particularly soil construction from waste materials, and how those constructed soils can be tailored towards the requirements of different communities of plants and used for habitat restoration at post-industrial sites. So I'm particularly interested in hard rock quarry sites. I did a lot of research in slate quarry sites in Snowdonia, and I'm also really interested in really nasty contaminated soils. I find those really interesting, and how we can clean those up using as little as possible energy through the process. And the Eden Project is a really amazing place to be able to study that, because I'm sure Emma will tell you a little bit more in her presentation in a bit, but there were 80,000 odd tons of soil constructed at Eden, because if you have a bare site and you're looking to restore it, you can either wait a couple of hundred or a couple of thousand years for natural processes. What you can't do is dig up intact soil from somewhere else and move it. So in the case of Eden, the soils were constructed from a mixture of mineral and organic waste materials. And that was just over 20 years ago. So there's a really amazing opportunity at Eden to study the behaviour of those constructed soils after 20 years of being cultivated. So that's my kind of research background. Now I'm responsible for this, and I look after these. So we have about 130 students who are studying plant science degrees, garden design, and as of the last 2 years, we also have our master's degree in ecological restoration. And the projects that our students are doing over the summer fit into the bigger picture of the activity that's going on in Cornwall and the rest of the world. But it's the projects that we're involved with that I'm going to focus on for this presentation to tell you what we're up to during our Raucous summer. Firstly, I talk about ecological restoration. And so what's the difference between ecological restoration, ecosystem restoration, conservation, rewilding? How do they all fit in? So a couple of definitions for you, one from the United Nations and one from our professional body, the Society for Ecological Restoration. And ecological restoration is the practice which is underpinned by the theory of restoration ecology. So a lot of the research that we're doing is to add to our understanding of restoration ecology to allow practitioners to use that information to successfully restore ecosystems. And by restoring ecosystems, we're talking about how people can assist with the process of recovery in a system that we have damaged, degraded, or completely destroyed. And ecological restoration aims to establish an ecosystem that can be resilient and respond to change, and is on a trajectory towards full recovery. And it's important to say that we're placing it on a trajectory towards full recovery because we haven't had scientific method for long enough to study the process of the development of many ecosystems. We certainly don't know the full biodiversity in many ecosystems, particularly once you get below ground and into the soil. So there is no restoration ecology, there is no ecological restoration project that we would consider to be finished and complete. But what we're doing is we're trying to engender the conditions where an ecosystem can develop and reach some degree of self-organization and resilience. And it's also important to say that it's not a substitute for conservation. So in conservation, we might be seeking to maintain populations of interesting or scarce species. And in many instances, and there's some relevance here to what Maria will talk about, that might mean that we want to intervene and stop natural processes occurring. We might want to prevent the natural succession from heathland to scrub and into woodland because we value some of the species that are found at those earlier successional stages. And it's also important to say that we have changed the abiotic conditions, the non-biological component of the planet, massively. So we've changed particularly the amounts of nutrients on the planet, nitrogen and phosphorus. And conservation now to maintain particular populations might require different management practices to the management practices that were needed 10, 20 or even 5 years ago. So one of the differences between restoration and conservation is we still need conservation, but we also need to recognize that the biosphere is in an impoverished condition. We're responsible for a mass extinction, and what we're trying to conserve now is a level that's very far below the level that we might have expected it to be 5, 10, 100 years ago. And the point of ecological restoration is that the concept that we can actively improve our environment rather than just sustain it at that impoverished condition. So that's where the optimism comes in. Genuinely, without exaggerating, I've had students come across the car park at Eden to me close to tears and say, this is in the news, this report has come out, we've only got 10 years left to save the planet, we're all doomed, aren't we? What's the point of spending 3 years doing a degree? And ecological restoration is an antidote to that, because we're saying, yeah, we recognise we've had a massive negative effect on the environment, but here are these ways that we can improve our environment, and here are these case studies that we would consider to be successful. And we need you to learn the techniques to be able to restore ecosystems appropriately. So that was too long, but it's important to note that to restore an ecosystem, we need to be aware of our societal impacts. We need to halt and reduce the causes of degradation in a system to start with. So that might be the eutrophication of the system that I've talked about, the wastage of nitrogen and phosphorus, particularly from agriculture. We cannot restore ecosystems effectively unless we can stop that problem at source. It might be the discarded fishing gear around the coast of Cornwall and how that entangles marine mammals. So we need to halt those causes of degradation and reduce the impacts that we have as society on ecosystems. And then we can arrange things so that we're improving the way that we interact in ecosystems and that we put them on a trajectory to being recovery. And quite a good way of looking at it is, why do we need to intervene? Do we need to intervene, or can we just let nature take its course? But it depends on the degree of damage, and it depends on the type of ecosystem. It depends on the proximity of that ecosystem to more functional systems for sources of seed and for animal migration. And I quite like this chart because it makes a distinction between physical and chemical barriers that we might refer to as being abiotic, non-living, and biological barriers. So an abiotic barrier on agricultural land, if we think about cauliflower fields, I have a particular pet hate of cauliflower fields around me, and the massive declines that we've experienced in soil organic matter in those kind of agricultural systems. If we want to restore those to more biodiverse systems, then we need to add organic matter back into the systems. If it's a contaminated former factory site, then it might be that there are metals and organic contaminants that are preventing the ability of that ecosystem to recover. And if it's a river, it might be that we've dammed it and we've prevented the natural migration of fish. So if we're looking to restore an ecosystem, we first have to look at the causes of degradation, then we have to look at the physical and the chemical barriers for ecosystem recovery. And once we've addressed those, in some cases, an ecosystem will have the ability to restore itself. If it's close enough for animals to migrate or to sources of seed, then it might be that we don't need to do very much else. But it might be that those species aren't present anymore and need to be reintroduced. It might be that there isn't a source of seed and we need to plant trees. Or it might be that the native vegetation is being outcompeted by the invasive species like Japanese knotweed and rhododendron and Himalayan balsam, and that the ecosystem can't recover to reach its biodiversity potential unless we address those. So we look at those biotic barriers, and we plan how we're going to deal with those, and then on into the longer term, to maximize biodiversity and the services that that ecosystem can perform for us, we need to look at how we manage that ecosystem. With the increase in nitrogen on the planet, do we need to cut our wildflower meadows more frequently? Do we need to reintroduce controlled grazing or browsing and burning? So all those elements of management that we need to put in to further improve the condition of an ecosystem along that continuum. So our work in doing that is informed by international standards and best practice. So we recognise the need to engage with stakeholders, to draw on lots of different types of knowledge, to identify suitable reference ecosystems, and that forms pretty much a whole module on the course. Is actually, we have this site what could it become, who gets to decide, and how do we set our success criteria. And then we recognize that if we can connect this activity together, then we gain cumulative value when we apply these techniques at large scale as part of a continuum of restorative activities. What's the difference between that and rewilding? Actually, not that much. I would personally, and there can be a lot of debate about this, I would consider rewilding to be one aspect of ecological restoration at one end of a continuum, from conservation through to rewilding. There's a lot of debate in the literature about this. It's an unpopular term in many scientific circles, but it's inspired a lot of people. So personally, I think it's one that we should be using, but we need to define what we mean by rewilding. Otherwise, we risk the danger of people assuming that means we're trying to remove people from the system, which isn't the objective in rewilding projects. So I'm just sharing that the 10 principles developed recently by the International Union for the Conservation of Nature that explains how rewilding is about restoring the interactions between organisms and their environment in ecosystems, using the concept of the 3 Cs, the core areas, the connectivity and the coexistence with people, and then through to the end that it does require a paradigm shift in the way humans and nature coexist into the future. So I said it's going to be a busy summer. June is particularly busy for many reasons, not the G7, but the 5th of June is Earth Day today. It's Environment Day on the 5th of June, and that's the official launch of the UN Decade on Ecosystem Restoration. So it's a decade of intense activity to try and revive the health of our ecosystems, culminating in the deadline for achieving the UN Sustainable Development Goals, and roughly corresponding with the timeline that us scientists think is the last chance for preventing catastrophic climate change. So June is very, very busy. And the next 10 years need to be very, very busy. And I nicked this slide from a presentation we had recently from Project Seagrass. We just all really liked it because these are the 17 UN Sustainable Development Goals, but nested to show how we can only achieve our societal and economic goals if we can invest in restoring our biosphere, because we need a healthy environment to achieve all of the rest of the goals. And we would go further in our particular group and say that a particular component of the biosphere that has been under-researched and underfunded is soil. And without healthy and functioning and biodiverse soils, we can't establish the vegetation and the wildlife, and then we can't achieve the levels of welfare that we want in our society. So on to some of the projects. So we're not doing it in isolation. We have 15 projects, and almost all of our projects have an industry or an academic collaborator within them. So we're working with lots of different wildlife trusts. We're working with Cornwall Seal Group. We are working with the Society for Ecological Restoration and some big employers like Imerys. And I just want to give you a flavour of some of the kind of project activities with some pretty pictures of things that we've been up to over the last couple of weeks. Actually, most of these pictures are. So this is Cabilla in Cornwall on Bodmin, and Cabilla is at the beginning of a rewilding, although they're not that keen on the term rewilding. They prefer restoration, but certainly reforestation. So Cabilla has been a mixed farm, has got some outstanding patches of ancient woodland that have been protected from agricultural intensification because of the rockiness and the topography of the site. It's an Atlantic rainforest, so it's a really interesting oak woodland habitat, and they're looking to rewild about 80 hectares of Bodmin to restore connectivity between patches of woodland and to plant lots more trees on agricultural land in the area to do that. It's a really, really beautiful site. I think it's Cornwall's only, or certainly one of the few, wooded tors in Cornwall, surrounded by some ancient hut circles. And these pictures were taken last week, and it's just starting to come into life now with all the understory plants and the bluebells. This is our student Liz, and my 10-year-old Barnaby, who was being our field technician for the day. And one of the projects that we're working on there is to do some paleo ecology. So we know what it looks like now. What did it look like several thousand years ago, and what could it become? So the work that we're doing there in terms of that kind of baseline analysis is to take some peat soil cores from the wetter areas of the site. So we're looking at potentially a couple of thousand years of soil development in the core there. And then they will be analysed in Plymouth's paleoecology labs to look at the relative proportion of material from grasses and trees. So doing quite a lot of pollen analysis to say a couple of thousand years ago, was it closed canopy woodland, was it grassland, was it wood pasture? And then we have the narrative from the site, hopefully back kind of 9,000 years to the current day, and then thinking about what it could become over the next 1,000 years. We've got beavers at Cabilla, so we've got Jean-Claude and Sigourney, the beavers in an enclosure there. And another project that we've got is looking at how the beavers affect birds. So we're really lucky in the southwest in that we've got beavers all over the place. So we're able to compare a couple of different beaver enclosures and look at how the changes in landscape and the presence of the beavers affects the diversity and the behaviour of the birds in the enclosures. We're working with Imerys at Raucous Downs, which is one of Cornwall's oldest and most extensive heathland restoration sites that was done back in 1997 on an area of China clay spoil. And as you can see, it is scrubbing up. It's also been invaded by lots of invasive species like rhododendron. So our students are working at Raucous Downs to do the soil and the vegetation survey and put that onto a map database, which we can then give to Imerys, and that will help to inform the level of management on that site. Whether we want to keep it as a heathland, whether we want to use it to restore fragments of heathland, or whether actually it's better to let it scrub up in terms of carbon storage and habitats for birds. We also have the Eden project is kind of associated with the biomes, but what we also have on site is a rare wetland habitat. We have a rain-fed, what's called an ombrotrophic bog, and this type of habitat, or these particular soils, are particularly important for carbon storage in the UK. It's one of our biggest stores of carbon is in the soil. It's more than 80% of our carbon stocks globally, and even more than that in the UK because of the rainfall and the types of soils we have. It's also a really valuable habitat for reptiles and amphibians and some interesting plants and insects. So this was us yesterday doing some measurements of carbon dioxide emissions from the soils on the site. So the site is drying out. You can see the sphagnum around here, and as it dries out, the decomposition of the organic matter stored in the peat starts to accelerate, and what we get is big emissions of carbon dioxide. So this graph here is showing increase in carbon dioxide concentration in a chamber connected to this gas analyzer, and we can use that to work out how much carbon the site is emitting, and hopefully we can re-wet it to try and keep it at a wetland state and prevent some of those emissions of CO2. So we're looking at how we can potentially bund the site and block drains with the brash from the scrub clearance to maintain it as an interesting bog type habitat. We are also working with the University of South Bohemia in the Czech Republic as part of a European-wide project where we're surveying sites at different ages since abandonment that haven't had any active intervention, and then we're looking at what develops naturally. And that information is going into a European-wide database, so land managers can say, if I don't do anything, there's a high probability that this vegetation community or this depth of soil will exist on this site in 5, 10 or 50 years into the future. When he presented his research proposal, Julian included lots of pictures of dog poo. I've toned it down a little bit and nicked this picture from Dynamic Dunescape. So Julian is spending his summer looking at the impact of dog poo on duneland ecosystems, because it's potentially a significant source of nutrients like nitrogen and phosphorus into the dunes, and perhaps if we can give that information to people as well as the standard information that we give them, we might have be able to influence behaviour. And then two more projects to tell you about really briefly before I hand over to Emma and Maria. We've just been awarded a share of a 2.5 million euro project with Plymouth and 5 other industrial or academic partners, and that's for a project called Recon Soil. The logo hasn't officially been released yet, so I'm being a bit naughty putting it on there. But this project is designed to improve practice in the construction sector, particularly so that we send less soil to landfill. We treat soil as a resource, and through this project, what we're doing is developing new soil mixtures and blends with organic waste materials for land restoration and landscaping, and the slightly more controversial bit for horticulture as well. And also with the connections through the Cornwall College Group, we're looking at developing a significant amount of training, for example, for modules that could be taken by apprentices in construction and engineering to teach them more about the value of soils and how to manage them on active sites. And then finally, just to end with a nice picture, so we've recently also been awarded funding from the Royal Society through a partnership grant, which is to work with Kernow Learning Academy and 4 primary schools around Eden on a project called Understanding Biodiversity Conservation. So that project is aimed at teaming up Year 4 pupils with master's degree students and getting them to learn more about how to assess their school grounds for biodiversity and how their grounds make them feel, and make plans on how they can improve the environment around their schools. So we're looking forward to welcoming quite a few coach loads of Year 4 kids to Eden for our MSc students to do a little bit of teaching. Maria and Emma probably don't know the full details of that one yet, but you will be involved with that one and it'll be a lot of fun. Okay, I'm going to stop at that point and stop sharing my screen. So that's a brief rattle through some of the projects that are going on. But if I could hand over to Maria and Emma now to give us a bit more detail about what they're working on. Who wants to go first?
Right. So hello, my name's Emma. So I'm here to give you an overview of my project, but also just kind of to talk a little bit about using environmental DNA for biodiversity assessments, which is what my project is related to. So I suppose just to start off, sorry, mine seems to have frozen a bit. Right. So just a bit about me. So I'm kind of in the process of a career change, as Maria is, because we'll both kind of tell you this. So basically, I've been working the last 18 years as a nurse, but recently completed a foundation degree in horticulture at Eden Project Learning. And whilst I was studying on that course, I developed an interest in ecology at kind of a more landscape scale and also kind of ecological restoration. So I've been given the opportunity to study on the master's degree with Mark at Eden Project Learning, and due to finish that at the end of August this year. What is environmental DNA? I think this picture is pretty good at summing up the fact that we're taking samples from the environment and looking at the DNA of different organisms within that, and that's kind of made up of everything basically. So plants, animals, invertebrates, fungi, bacteria, kind of got it all in there. All living things have DNA. Environmental DNA, or eDNA, is released from an organism into the environment. It comes from the soft cells, so like skin, hair, roots, degraded cells. We get that from the environmental samples we can take from the environment. So soil and water, the most common that we would type of environmental samples we would take. We're looking for specific regions of DNA which are unique to specific organisms, and these can be used as barcodes to help identify just millions of organisms. And there are big databases, kind of open access online, to help kind of ID the organisms. So it can be a really good way to detect different organisms in a non-invasive way. So, for example, with invertebrates, to do an invertebrate survey, quite often you'd have to catch, sometimes kill the animal or the invertebrate to ID them. So obviously being able to take a sample of soil or water, for example, is much nicer on kind of the organisms in the environment. So how we can actually use environmental DNA, kind of detecting rare or elusive species. So quite a fun example is the Loch Ness Monster. So it's kind of a recent project by a university in New Zealand, kind of looking at all the different organisms within Loch Ness to see if there actually is a Loch Ness Monster. Unfortunately, it didn't show the presence of any new species, but it just kind of can show you how you can use eDNA. Invasive species monitoring is another really common use of environmental DNA. So signal crayfish is a really problem species in the UK, and that's often monitored kind of looking for its DNA in water, kind of aquatic environments, so looking at water samples. Other things are monitoring species changes over time and kind of the actual community makeup of species in an environment, but these are seem to be restricted mostly to kind of use within research at the moment. So not necessarily widely used in kind of ecological practice. So the use of eDNA for biodiversity assessments is something that I'm looking at for my project. And just to kind of give that a bit more context, it kind of started off with talk we had from Dr. Julian Donald, who's a postdoctoral researcher at the EDB lab in Toulouse University in France. So EDB in French is the Evolution and Diversity Biologique lab. So they've been researching sections of rainforest in French Guiana, conducting species inventories over large areas, looking at community composition of different plants, also animals and invertebrates in these areas. And what they found was there's still quite a few unanswered questions with using kind of the environmental DNA method for biodiversity assessments. Some of them are kind of, we don't really know how long DNA stays in the environment after organism is gone. You know, what actually affects kind of DNA degradation in like soil. How does it respond to temperature, humidity, soil chemistry, all of those questions. Do all organisms shed DNA in the same manner? We just don't really know at the moment. And is the signal of individual plants lost in areas with higher diversity? So if you've got so many different plants or other organisms, you know, do you lose the signal of those single plants? And how far does the signal travel in the environment? So if you're taking a sample, 40 miles away, does that pick up everything in a 40-mile radius or just within a 10-meter radius? And that's something we're still kind of trying to find the answers to. So that's kind of where my project comes in. So the researchers at the EDB lab chose to take some samples at Eden Project, just because it's a more controlled environment. As Mark was talking about, it's created with constructed soils. It's within a semi-enclosed environment with kind of very little they have traffic going through, but they have kind of biosecurity controls in place to restrict kind of plant material and other things coming inside. So what Julian Donald and kind of his colleagues did is take some soil samples and analyse them for plant DNA from sites in the Mediterranean and the tropical biomes. And what they weren't able to do at the time is actually compare this to the plant species that are in situ in these sites, and that's where my research is coming in. Because do we know if this is an accurate inventory of the plants present in the biomes? Just because the DNA is there, does that mean that the plant's there, or does that mean that someone's kind of walked in and brought a leaf with them from outside, and actually it's that DNA that's kind of sitting and that we've managed to collect in the soil? And that's what I'm going to be looking into for my projects, actually starting next week. So kind of taking advantage of the Eden Project being closed at the minute to visitors is kind of myself and Julian are going and take these, do some more sampling. So my research is focusing on on what's above ground. So conducting the plant surveys from the sample areas where the DNA was taken from and comparing these with the DNA results that Julian already has. Also looking at root biomass, so actually taking some soil samples from the DNA sampling sites to see if there's any relationship there between kind of the amount of roots the plant has as to the amount of DNA that has been picked up from the samples. But also measuring environmental conditions in the soil, so looking at pH, rate of respiration, so kind of how quickly the DNA might be breaking down in the soil, and also the organic matter kind of content of the soil as well, just again to see kind of what relationship this does have with the different signals of DNA and kind of how strong it has been. And so what I think for me, what my interest is and kind of what my hope is that then that eDNA can be used for biodiversity assessments over really large spatial and temporal scales, that that we can then start monitoring or monitor better kind of restoration and conservation projects. As Mark said, we don't know enough about the trajectory of ecosystems. We don't know what's going to happen, but actually if we can sample large areas and look at all the organisms in these areas in a quick and easy way, so by looking at the DNA in the environment, then we can have a much better idea of how ecosystems are developing and what factors affect that. With the technology of DNA developing, we can potentially do quick and easy assessments in the field, and then just do much longer term monitoring of restoration, which can then inform research and practice. So obviously it's monitoring, and I've put on the end here, monitoring all known organisms and then potentially discovering new ones. We have DNA records of organisms we know, and potentially find new ones because of this, really. So yeah, for me it's a technology with quite an exciting future. So yeah, it's just to say thanks to obviously Eden Project for letting me do it, and then Eden Project Learning for giving me the opportunity, and Dr. Julian Donald, who I'm kind of working with on the project. Yeah, thank you.
There we go. So hopefully you can all see that. So I'm going to speak to you today about storing carbon and forming soil organic matter in a temperate wetland on the Isles of Scilly. So firstly, same as Emma, a little bit about myself. So my first career was as a tax solicitor, but in 2019 I quit my job and joined an organisation called On Purpose, which is a career change programme focused on roles in impactful organisations. This year really fueled my passion for the environment, and while studying an online course in ecosystems restoration, I knew I wanted to learn more because I understood how restoration could be a really positive action to address global issues. So this has brought me to the Eden Project and onto this master's programme. So my project is based at Lowermoors. So that's it's a freshwater temperate wetland and a site of special scientific interest on St Mary's and is managed by the Isles of Scilly Wildlife Trust. It was designated as an SSI due to its presence of freshwater plant species, and these are the type that you would expect to find on a fen or on marshy grassland. Lowermoors is also extremely important for St Mary's, as it provides a proportion of their drinking water. However, seawater has been infiltrating the site for a number of years, and this has resulted in changes in both the plant communities, but also raising concerns about protecting the drinking water. So one of the pieces of information which the Wildlife Trust would like to know about Lowermoors is the level of carbon which is sequestered on the site. So my project will focus on the carbon sequestered in the soil as soil organic carbon. So plants are the main contributor of carbon to the soil by depositing both above and below ground biomass through sort of litter, roots and root exudates. These deposits are decomposed by microorganisms within the soil, and carbon is stabilised within the soil when it's protected within stable fractions of the soil organic matter. This protection prevents microorganisms from accessing and mineralising the carbon. So it was previously believed that soil organic matter was predominantly composed of undecomposed complex plant compounds. However, new soil analysis techniques have produced evidence that the most stable soil organic matter contains mostly microbially derived products. So it has been theorized that the simpler, faster decomposing plant compounds, which are easier for microorganisms to use, are actually more important for soil organic matter formation than the complex plant compounds. However, as science continues to kind of develop and move on, more recent studies are advocating for a more balanced approach and indicating that there are actually likely to be at least two pathways for soil organic matter to form. One results in these microbially divide inputs, and another enables plant-derived residues to be incorporated directly into soil organic matter without first having to pass through any living microorganism. So my project is going to consider the effects of both soil salinity and plant compounds on the formation of soil organic matter and the stabilisation of soil organic carbon at Lowermoors. I'm really hoping that my results will show that the areas of the site that have the highest levels of soil salinity will also have the vegetation communities with the highest levels of complex compounds, and that these areas also have the lowest levels of soil organic carbon and soil organic matter. As you can see from the map on screen, there are 7 different vegetation communities identified at Lowermoors, and I'm hoping to be able to make comparisons between these. So in June, I'll be travelling to St Mary's and spending 4 days with the colleagues at the Isles of Scilly Wildlife Trust to collect samples. I'm planning on taking 2 soil samples and a vegetation sample at 5 different sites within each one of the 7 identified vegetation communities. The reason I'm taking 2 soil samples is that I want to be able to consider the difference between the levels of soil organic carbon and soil organic matter at different soil depths. So to date, soil science has focused on the top 10 centimetres of soil, because this is relevant for agriculture and it's the part of the soil most active for plants. However, it's becoming increasingly clear that what is happening deeper in the soil is really quite relevant for carbon and nutrient cycling. Once I've collected my samples, I'll be using the lab at Eden to analyse soil samples for salinity levels, measuring the electrical conductivity. I'll also be measuring pH, as this is understood to affect the microorganisms' activity within the soil, and using gravimetric testing for total soil organic matter and total soil organic carbon. I'm also really hoping to use the labs at Rothamsted Research in Devon to undertake fibre analysis, and this will help me to understand the levels of the complex plant compounds in the different vegetation samples. Once I've collected all my data, I'll be able to interpret this and pull together my findings and provide the Isles of Scilly Wildlife Trust with some management recommendations. So I'm really hoping to be able to present both practical and theoretical outcomes to my project. Practically, this project will provide the Wildlife Trust with its first data set about the levels of carbon stored at Lowermoors. And then this data and the management recommendations that I put together can be incorporated into the future management plan for Lowermoors, which the Wildlife Trust is currently writing for the site. Internationally, many freshwater wetlands are experiencing increasing levels of salinity and will be experiencing changes on their sites which could be quite similar to those we're seeing at Lowermoors. So therefore the management recommendations for Lowermoors may be relevant and applicable to other sites elsewhere. In terms of theoretical outcomes, the soil organic matter theories that I was discussing earlier have been considered in wetlands in very limited circumstances. So therefore, this project should contribute to our understanding of their applicability in these ecosystems. Further, literature regarding the effects of soil salinity on soil organic carbon is ambiguous. Therefore, this study will contribute further data and understanding to these areas of study. And finally, wetlands are considered to be important in terms of climate mitigation. Therefore, understanding how carbon is stabilised in wetland soils and how to support and encourage this will contribute to scientifically robust nature-based solutions. So I just want to say a big thank you to the Eden Project Learning and the Isles of Scilly Wildlife Trust for supporting me in this project, and thank you all for listening.
Very good. Can I, yes, so what I would like to say is a massive thank you to all three of you, because that was incredibly interesting and really very important to our environment and, you know, the way we manage things in the future. And you know, some of the things that Mark said were very poignant at the beginning about, you know, the concerns we have about degradation and what we've done already and what we can be doing to turn things around pretty rapidly if we put our minds to it. Does anybody have any questions? Because unfortunately I don't seem to have access to a chat button on from Teams from my office, so I don't know if anybody's posted any questions. Colin, has anything come up as chat questions? No. Does anybody have any particular questions that they wanted to ask our very knowledgeable speakers? No. Well, can I just say a massive thank you to everybody for attending. Again, thanks to Mark and Emma and Maria, because loads and loads of information there for us, and keep up the good work is what I would say, because it's incredibly interesting and it's something that we should all be focusing on for future sustainability. So it's really, really good to have you all here and, you know, engaged with these activities. So thank you.
Thank you very much. Thanks, Carol. Thank you, Maria. Thank you, Emma.
Thank you.